All right, hello. This is chapter 12.1. So, this is the first day of DNA and the lecture into DNA. There's not a lot of notes to take today because there's a lot of black backgrounds. Remember, we don't take notes on the black backgrounds here. So, 12.1 is a history of DNA. And up until the early 1900s, we knew that cells contained chromosomes, but we really didn't know what chromosomes were made out of until the early 1900s. And we figured out that they were made of DNA, but we really didn't know what DNA was. We knew some of its components, but we just didn't know a lot about the structure of DNA. We also understood that the DNA is responsible for how traits and just inheritance patterns are passed from parent to offspring. We just didn't know how they were passed. At this point, scientists are trying to figure out who or what the structure of DNA is because they know it's an essential molecule and it's in all cells, and it's going to be really important going forward. So it's like the I don't even know how to describe it. It's like the thing we want to figure out. If you figure out the structure of DNA, man, you're going to be known in every biology textbook going forward. So at that time, we have a scientist named Rosalind Franklin working as a PhD. She had her PhD, but she was working on the structure of DNA. She was one of those scientists working to figure that out. You can see what Rosalind Franklin looks like. And she was using something called an X-ray crystallography machine, which takes, I have in quotes, quote unquote, pictures of really small things. And it does this by shooting a particle or a beam of particles at a small structure. And how they bounce off tells us a lot about that structure. I know that sounds really crazy and just really hard to understand, but understand these pictures help us see very small things because you can't see DNA under a microscope. It's just too small. She took photo 51, which is the evidence that we used at that time period for the structure of DNA, which is in the double helix. Let me show you what that looks like. So photo 51 looks like this. I know it looks like just a bunch of dots on a circular picture, but these pictures took hours and hours and hours to develop. And I know it doesn't seem like a lot, but as soon as we got this picture, we really understood, we could understand a lot more about the structure of DNA. Now, what actually happened was Watson and Crick were given her picture. The picture was stolen from her office without her even knowing. And that X pattern told them that the structure of the DNA was in the form of helix. Now, they were model builders. They weren't the science like the scientist that Rosalind Franklin was. She was more of a like data person where she took a lot of data and pictures and did the actual science. These guys were using data and trying to figure out the model or the structure of DNA. And they did build the first accurate model of DNA, but it was because Wilkins here, who was working with Rosalind Franklin, stole that photo without her knowing, like I said. So what that means is that these two are actually recognized as the fathers, we like to say almost, of DNA, or the people who are known as the who discovered the structure of DNA. Even though Franklin was the one that came up with the data, and you might say, well, why isn't she uh, recognized with this? And the sad news about this is these gentlemen were given the Nobel Prize for Physiology and Medicine back in 1962 for the discovery of DNA, but Franklin didn't get the award. And you might ask why. She had passed away in 1958 due to cancer, and they think it might be because of the X-ray crystallography machine and how radioactive it was. There were actually several times where during her work and during her studies, she was so radioactive she was sent on vacation because there was so much radioactive materials in her at her work that she just was so radioactive. They said, no, you're too radioactive, you need to go on vacation. So she died before they awarded the Nobel Prize, and you can't be awarded the Nobel Prize posthumously, which means after you have died. So she was never given credit for her discovery. All right, so that's the end of chapter 12.1. Now we're going to get to chapter 12.2. So this is the second day of notes. So we're going to go over something called DNA, which you've probably heard a lot about before, maybe even studied a little bit before in your middle school classes. But DNA stands for deoxyribonucleic acid. I don't really want you to 100% be able to like spell this out, but I do want you to understand this is what it stands for. It's not just a molecule. It actually stands for deoxyribonucleic acid. And that's what this molecule here looks like. And DNA is the blueprint for your proteins, the blueprint for what makes you you. I always say this is who you are. This is your genes. Now, not everything is going to be in your genes. So like things like personality and sometimes how tall you grow, things can be environmental as well, but this makes you who you are. And DNA, you can see, I love this stat here. A single human cell contains more than 2 meters or 6 feet of DNA stretched out. So DNA is made up of something called a nucleotide. Nucleotides have three different parts. They have a phosphate group, they have a sugar molecule, and they have a nitrogenous base. Wow, that's a lot of words here. Okay, first group, phosphate group. That just is represented by a PG or a P. That's just the phosphate. Two, sugar molecule. That's this right here. That's your sugar molecule. This is a deoxyribose. You can tell this is a sugar molecule because it says deoxyribose, the OSE at the end. And then last year it says nitrogenous. That just means it has nitrogen. So we have a nitrogen base. This is just some molecule here that we also have, and it can be either a single or double ring. This will make more sense as we look at a couple different nucleotides. But you can see the phosphate group, the sugar, and the base. Now, DNA is made of long chains of these monomers, or these long chains of nucleotides. You can see this is a DNA molecule here because we have long chains of the nucleotides. And there's 4 different nucleotides. You can see adenine, guanine, thymine, and cytosine. So we have 4 different nucleotides, but most of the nucleotides are going to be exactly the same. You can see here the phosphate group and the deoxyribose group are going to be exactly the same each time. It's the A, G, C, T. It's the base part that's different. That's what makes a nucleotide different. Pyrimidines. So within these nucleotides, you have 2 different types of nucleotides. You have pyrimidines and purines. You have pyrimidines, which have a Y. I always remember that because cytosine and thymine also have a Y, and they are single rings. So you can see here we have our pyrimidines. These are cytosine and thymine. Purines, we see they have double rings. The purines, think adenine, guanine. So cytosine, thymine are pyrimidines. Purines are A and G, adenine and guanine. If we look back here, if we go back to this previous slide here, you can see our 2 purines, which have the double rings, and our 2 pyrimidines, which have the single ring. Okay, looking forward here. So purines, pyrimidines. What you're going to notice is each purine is going to bond specifically to another pyrimidine. You can see this single thymine is going to bond with this double adenine. So we have a single ring here, a double ring here, adenine. So our A's and our T's go together. Our C's and our G's are also going to go together. You can see here our C as a single ring, our G as a double ring. They're going to bond together. Now, with this DNA base pairing, you can see again, A's and T's bond together, G's and C's bond together. You can see that this molecule has the A's and T's, G's and C's bonding together. I always remember G's and C's being like the curve here. Yeah, and then the T's and A's, yeah, they go together as well. Okay, so I usually, when I'm doing this in class, let me go back here. I usually have the students tell me what each of these is going to be. So you can see here like A, G, or the A, G, C, T. It's going to go T, C, G, A, and so on and so forth here. And I have the students tell me what's on the other side here. And I give them this good meme at the end. So I'm sorry, G, but we're basically not a good match. It's because A and G don't bond together. All right, 12.3 is going to look more specifically at the structure of DNA and how those nucleotides are going to bond together to form what we know as DNA, the double helix. So remember that we have these nucleotides, and they're actually going to bond together to form 2 chains, and we call this a twisted ladder. It's a twisted ladder of nucleotides that forms this double helix. Now, these nucleotides are on the sides here are held together by something called a covalent bond. We talked about covalent bonds a little bit when we talked in chemistry. These are very strong bonds. So these bonds you can see between these sugars and phosphates on this side and on this side are known as covalent bonds, and they're very strong. We want those to be very strong to keep the DNA molecule secure and together. Now, we also have something known as the sugar-phosphate backbone. That is what we're just talking about, where we have the sugars and the phosphates held together by a covalent bond here, and that's our sugar-phosphate backbone. That helps support the nitrogenous bases, which are our most important part of the DNA molecule because that carries our information. Between the nucleotides, we have things called hydrogen bonds. You can see here between each of these bases, we have hydrogen bonds. Between guanine and cytosine, we have 3 hydrogen bonds. Between adenine and thymine, we only have 2 hydrogen bonds. So you can see we have hydrogen bonds between the bases and then covalent bonds between the nucleotides. Now, I kind of make this analogy with the DNA molecule that they run in opposite directions. So you can see we have the phosphate group start up here, and we end with a sugar molecule. But on this side, we start with a sugar molecule, and we end with a phosphate group. This is known as, and you can see I always relate this to like the pathway on a road. It goes in opposite directions. This is known as being anti-parallel. And we give the notation of 3' to the phosphate group and, I'm sorry, 5' to the phosphate group and 3' to the sugar. We're not going to go into why this is, but just remember this as the 5' and 3' end. So 3', 5' end, they run in opposite directions. So remember, this is just a good recap of DNA. So nucleotides make up DNA. DNA makes up chromosomes, and you have millions of nucleotides inside a single chromosome. Chromosomes, there are 46 of them, are in each one of your cells, and each chromosome consists of over 3 billion base pairs of nucleotides. That's a lot of nucleotides per cell. All right, Chargaff's rule. This is the idea that the idea is not the greatest word there, but the idea that the amount of adenine and the amount of thymine are going to be equal, and that should make sense because the amount of adenine is going to equal to the amount of thymine because they bond together. So that for every adenine, you have to have a thymine. For every guanine, you have to have a cytosine. So in this case, you can see here we have guanine, cytosine, adenine, thymine. They're equal amounts because they have to bond with each other. And this is true for all species. I go through these questions, and they're pretty easy. You find a new type of bacteria and sequence the genome and find the percentage of thymine is 12%. What's the percentage of adenine? That's easy at 12 because they're the same. This next question is going to be a little bit more difficult. So it says you find a new type of dog and sequence the genome and find the percent of thymine is 10%. What is the percent of cytosine? So it's not 10% because thymine bonds with adenine. So what you have to do is you have to take what's left over when you have, so you have 10% of thymine, you should have 10% of adenine. Because remember, adenine levels and thymine levels are the same. So again, thymine, adenine are the same. Both are 10%. Well, what's left out of the 100%? So you should have 100. Let's see if I do this the math here. Yeah, you should have 100. You minus the 20% because each of these is 10%. You get 80%. So your 80% should equal the C and G concentration. So you just got to take that divided by 2, and that's where you get your 40. We won't go through this in one more question. We'll just end up. And our next section is going to be DNA replication, or 12.4. All right, our last section here is going to be 12.4, which is on replication. This is where it can get kind of difficult because there's a lot of working parts here, but there's not too much new material. Like there's not like a ton just in this one section. So DNA replication, this is the process of producing 2 identical replicas from one original DNA strand. We talked about DNA replication when we were looking at mitosis. And if you remember, this occurs during the S phase. Remember that S phase where we're going to copy DNA? Well, and again, right here, let's go back. This S phase is where we're going to duplicate the DNA. We're going to make an exact copy. So remember, this is where we're at in the mitotic cycle, but we're going to be specifically looking at how DNA actually does that replication. So each strand is going to act as a template for producing a new complementary strand of DNA. I absolutely love this image here. This image does a great job of explaining this process. So we start out here with the 2 yellow bands of DNA. You can see how each one of them are going to be used as a template for the next strand of DNA. Then we're going to split those. So you can see we have a yellow, red, red, yellow. That's showing that each time we split, we're using each side as a template to make the other side. Remember, we're still bonding A's and T's, G's and C's together on each side, but we're using each old strand as a template for the new strands. You can see this bottom strand has all black DNA strands of DNA. You can see how it's called a semi-conservative model where the old strand is used as a template for the new strand. So the origin of replication. The origin of replication is the region on the DNA where the DNA is going to be initially replicated. So this is the start of replication. So right here, origin of replication. Next is helicase. So once we have that origin of replication, that's where DNA is going to be copied. It's going to start to be copied. This enzyme, we're going to be going over a lot of enzymes here. Helicase is an enzyme that unzips the DNA molecule, and this is what breaks up the hydrogen bonds. You can see here, this helicase is unzipping the DNA, and specifically, it's breaking those hydrogen bonds. The replication fork. The replication fork moves as the helicase moves along the DNA strand. Excuse me. So what happens is the helicase is breaking apart these hydrogen bonds, and that replication fork is that location where the helicase is doing that. So you can see here, as it moves along, it's going to move that replication fork as it travels down the DNA strand. I always say that the origin of replication is like where you're at when you start a story. So this is like the origin of replication. But as you're reading down through this story, that's your replication fork. So the origin of replication never changes, but your replication fork does. So you might be reading down here, that's where your replication fork is, but you start up here, which is your origin of replication. All right, DNA polymerase. This is the molecule, or this is the enzyme that is going to bond free-floating nucleotides to the growing chain of DNA. You can see down here, we have 2 DNA polymerases. They're going to bond these things called free-floating nucleotides. They're just random nucleotides in your nucleus, and they're going to bond together to form your new strands of DNA. Remember, what we're doing in mitosis is we're duplicating, and we're trying to duplicate the DNA so we can have enough DNA so each cell can split and have that DNA. So what we're trying to do is we're trying to make an exact photocopy of that DNA. And DNA polymerase is that enzyme that's going to bond the nucleotides together to make sure we have A's and T's bonding together and G's and C's bonding together. One important thing to remember is that this enzyme only works in a 5' to 3' direction. So what happens in our strands is we have 2 strands. First strand is known as the leading strand. The second is known as the lagging strand. This is the leading strand because the DNA is being made continuously, and it runs in the direction of the replication fork. The lagging strand up here is going to go away from the replication fork. The reason why it's going away from the replication fork is, remember, it's only going to travel in a 5' to 3' direction. So we can't travel in this opposite direction because of the anti-parallel nature of DNA. It has to make these small chunks called Okazaki fragments that will be fused together later on. But this lagging strand is what specifically is going to be one of the strands that is discontinuous. It's not made continuously. The leading strand is made continuously. So our end product of DNA replication is just 2 identical strands of DNA. And again, we need these 2 identical strands of DNA so we have enough DNA so when mitosis occurs, you have enough DNA for each cell. This is going to end the chapter on DNA structure and DNA replication. These are all the 4 days.